Hello and welcome back to another biology episode. Today we're talking about the cell cycle, which is a highly organized complex that determines the state of each cell within our bodies. We have a bunch of different proteins that act according to signals not only from the inside of the cell but also from the outside. If there are any flaws in this system, the consequences are disastrous, and malfunctions can lead to potentially fatal diseases such as cancer. The process of the cell cycle can be divided into 4 phases. So we've got the G1, the G stands for gap, the S phases for synthesis, the G2 phase, and M for mitosis. There will be a separate video about mitosis linked above. By undergoing this cycle, a cell grows and ultimately we've got 2 genetically identical daughter cells. Cells also have the possibility, after completing a division, to go into the so-called G0 phase, which is a quiescent state. Some cells in this quiescent state may enter the cell cycle again. They can differentiate or they may remain in this state. Examples are neurons, as depicted here, and they are then called post-mitotic. Now let's look at the phases in a bit more detail. In G1, the cell grows and the contents get duplicated. In S phase, the DNA is replicated. Now each of our 46 chromosomes has 2 instead of just 1 sister chromatid. In G2 phase, the growth continues, but actually it's not really known what happens in G2, so the cell obviously prepares for the cell division, but the exact mechanisms remain widely unknown. In mitosis, we've got 4 phases called prophase, metaphase, anaphase, and telophase, and then we've got 2 identical daughter cells after they're divided during cytokinesis. One of the most essential features of the new cells is that they contain the right amount of DNA. So therefore, the whole replication process is very, very important. As I said before, malfunctions would have potentially lethal consequences. So we've got different mechanisms that ensure that everything works as it should. Every time a cell completes a specific step within cell differentiation, so-called checkpoints or checkpoint controls are used to ensure that this process is not repeated again. Furthermore, they are able to block advance in the cell cycle if there are any problems or irregularities. And they are essential in the development of cancer, because cancer cells gain the ability to overstep checkpoints. Cells obviously have to respond to extracellular signals, but it seems that there is a specific time between G1 and a few hours before the transition of G1 to the S phase where they are responsive to extracellular signals. If a cell makes a decision to undergo a cell division, the cell no longer responds to those signals. So there's a critical point, which is called the R point, and here you can see where it is within the cell cycle. R stands for restriction. As soon as a cell has reached and overcome this point, there have to be really disastrous events to prevent a cell from completing a division. So therefore, this is a very critical time for the fate of a cell. And therefore, this again might be really important in cancer development, because R point deregulation could be part of the formation of various cancer types. Well, obviously there are events when a cell can retract into G0 again, but the vast majority of cells undergo cell division once they've passed the R point. This was a very general overview of the cell cycle. Obviously there are so many proteins and complexes involved that cannot be discussed here, but this should give you a good overview of the different phases. As I said before, there's going to be another video about mitosis, which explains these mechanisms in more detail. I hope you enjoyed this video. See you in the next one.